Okay, so we're finishing up lesson 2 today. We're talking about modeling inheritance. We're talking about complex patterns of inheritance. And so let's get started on page 418. Modeling inheritance. Have you ever flipped a coin and guessed heads or tails? Because the coin has two sides, there are only two possible outcomes, heads or tails. You have a 50% chance of getting heads and a 50% chance of getting tails. The chance of getting an outcome, like heads or tails, can be represented by a ratio. The ratio of heads to tails is 50 to 50, or reduced 1 to 1. So, reading check, what does a ratio of 2 to 1 mean? Well, the ratio of 2 to 1 means that the first outcome should appear twice as often as the second outcome. Plant breeders and animal breeders use a method for predicting how often traits will appear in offspring that does not require performing the crosses thousands of times. Two models, a Punnett square and a pedigree, can be used to predict and identify traits among genetically related individuals. First, let's look at the Punnett squares. If the genotypes of the parents are known, then the different genotypes and phenotypes of the offspring can be predicted. Okay, so what do you need to know in order to use a Punnett square? You need to know the genotypes of the parents. Okay, oops, got the wrong one there. The genotypes of the parents. Okay, so you need to know the genotypes of parents to use a Punnett square. Now, you could use take a Punnett square that's been filled in everything else but maybe a parent, and then you could go backwards with it. But generally, we don't do that. Generally, we use a Punnett square to predict the phenotypes and genotypes of the offspring. All right, so in order to start it, you need to know the genotypes. All right, so analyzing a Punnett square. Figure 7 shows an example of a cross between two pea plants that are heterozygous. Remember, hetero means different. So for pea seed color, capital Y, and capital Y, little y. So each of them have one dominant and one recessive gene or allele for the gene. So yellow is the dominant allele. All right, so that's why it's Y. Green is the recessive allele, the little y. The offspring can have one of 3 genotypes. They could be homozygous dominant or capital Y, capital Y. They could be heterozygous like their parents, or they could be homozygous recessive with little y, little y. The ratio of genotypes is written as 1 to 2 to 1. 1 to 2 to 1 ratio, and so we'll look at that. Because capital Y, or homozygous dominant, and capital Y, little y, or heterozygous, both represent the same phenotype. So if they have these genotypes, they're both going to be yellow. They're both going to exhibit the trait of yellow. So the offspring can have one of only 2 phenotypes, yellow or green. The ratio of phenotypes is written 3 to 1. Therefore, about 75% of the offspring of the cross between the 2 heterozygous pea plants will produce yellow seeds. So a 3 to 1 ratio means they have a 75% chance of being yellow and a 25% chance of being green. All right, so about 25% of the plants will produce green seeds. All right, let's go back and look at how we use this in a Punnett square. So a Punnett square, let me turn it around this side. A Punnett square can be used to predict the possible genotypes of the offspring. So offspring from a cross between 2 heterozygous parents can have one of 3 genotypes. So first thing you do, so if this is one parent is big Y, little y, another parent is big Y, little y, I want you to think about each of these columns and rows as a possible allele that's going into the gamete. Remember, a gamete is an egg or a sperm. So let's say up here we've got the egg or the ovum, and over here we've got a pollen sperm, right? We've got the male part. And so during meiosis, this genotype is going to split. One of these is on one chromosome and the other is on a homologous chromosome. And so those homologous chromosomes split. And so there are two different things that this egg can contain, okay? They could contain this dominant allele, or it could contain a recessive allele. Oh, wait. They say place male alleles on the top. All right, so we're going to change that around. Sorry about that. And we'll place the eggs over here. My bad, I didn't see that. All right, so let's say this is the sperm or the pollen, and this is the egg. All right. Anyway, so the sperm during meiosis or the pollen could contain either the capital Y dominant allele or the recessive. And so the same thing for the egg. It could contain the dominant or it could contain the recessive. And so when you know the genotype, you split that genotype in two, because meiosis split it in two. Okay, so one possibility is in the first column, one possibility is in the second, and the same with the rows on the side. All right, so once you've got that set, and those go on the outside of the boxes, then you can take whatever's on top and repeat it down. Okay, so this Y is rewritten like that. And then you take whatever's on top of this column and you repeat it down. And since it's a lowercase, we're writing it over on the right hand side. All right, and then you take this row and repeat it across. It goes in all of those. And you take this row and you repeat it across. All right, so now we have 4 different boxes of possible gene combinations. Now, it just so happens that 2 of these boxes are the same. Okay. This top one on the right-hand side got a dominant gene from the egg, from the egg over here, or right, and then the recessive gene, the recessive allele came from the sperm. Okay, so this one at the bottom got a dominant from the sperm and a recessive from the egg. All right, so those 2 are the same. They're both heterozygous. Then we have, let's find a different color. Then we have a homozygous dominant here, and then we have our third type, a homozygous recessive here. So so let's take a look at that. What does that mean for the genotypic ratio? So for the genotypic ratio, it's going to be this one only appears once. This one, a little bit darker blue, appears 2 times. And this one appears once. So it is a 1 to 2 to 1 ratio for genotypes. Remember, the genotype is just the alleles that we write as letters. Okay, but what does that mean for their characteristics that you see? That's your phenotype. So remember that when you have at least one capitalized or dominant allele, it expresses the dominant phenotype. It expresses the dominant trait. So all of these 3 are going to express the dominant trait. And you can see they're all the yellow. And so that's 3 of those. But here we have 1 that expresses the recessive trait. So the phenotypic ratio is going to be 3 to 1. Okay? So visual check. What phenotypes are possible for P offspring of this cross? They might be yellow or they might be green. Those are the phenotypes that are possible. All right, let's go on to the next page. We're on page 419, using ratios to predict. So given a 3 to 1 ratio, just like we saw, you can expect that an offspring from heterozygous parents has a 3 to 1 chance of having yellow seeds. But you cannot expect that a group of 4 seeds will have 3 yellow seeds and 1 green seed. This is because one offspring does not affect the phenotype of another offspring. In a similar way, the outcome of one coin toss does not affect the outcome of other coin tosses. And you've seen this before. Sometimes you toss a coin and you get heads like 10 times in a row, even though the ratio, the expected ratio for 10 tosses would be a 5 to 5 ratio, right? You would expect to see about 5 heads and 5 tails, but you might get 10 heads in a row. You see this with families. Even though there's a 50% chance of having a girl each time you have a child, you might see families with 4 girls and 1 boy. Okay, so more of the girls were born than expected. All right, so let's go on. So, however, if you counted large numbers of offspring from a particular cross, the overall ratio would be close to the ratio predicted by a Punnett square. Mendel did not use Punnett squares. Okay, so he didn't have that tool. He could see the ratios. He focused on the ratios of what of the colors and sizes and everything of all the different traits. So he looked at the ratios. Let's see. Okay. Mendel did not use Punnett squares. However, by studying nearly 30,000 pea plants, his ratios nearly matched those that would have been predicted by a Punnett square for each cross. All right, so we're going to learn more about Punnett squares, or we're going to practice using them as we go on throughout this week and probably early next week too. So pedigrees, another model that can show inherited traits is a pedigree. A pedigree shows phenotypes of genetically related family members. It can also help determine genotypes. In the pedigree in Figure 8, 3 offspring have a trait attached earlobes, and we looked at that in our launch lab, that the parents do not have. If these offspring received 1 allele for this trait from each parent, but neither parent displays the trait, the offspring must have received 2 recessive alleles. And think about that for just a second. If there is a trait in the offspring down here, like these have the trait, these 3 have the trait, the recessive trait, the attached lobe. Okay, if it's present in the offspring, but it's not present in the parents, then it's been masked in the parents. So if it's not present in the parents, but it's present in the offspring, it has to be a recessive trait. It has to be something that has been masked. Okay, so they must have gotten, each of these 3 must have gotten the 2 of the recessive genes from their parents. And so 1 each of the parents. So each of these parents must have contributed a recessive allele. The visual check says, if the genotype of the offspring with attached lobes is little u, what is the genotype of the parents and how can you tell? So again, if this lady has little u, and this guy has little u, and this guy has little u, okay, this we don't know for this girl and this boy what their genotypes are yet. I don't think we could even find out, because look, they'd have to have at least 1 capital U, right? At least 1 capital U. We don't know if they have if they had kids, and then we might be able to determine from that. But we don't know for sure, and here's why. Because both of the parents exhibit the dominant trait. That means they have at least 1 capital U. okay, we can infer, based on the fact that they have 3 children who have expressed the recessive trait, that they have each of them has passed on a recessive gene to those 3 children. So these must be recessive. They must be heterozygous. Now we don't know about these 2 who have the unattached lobe, the dominant, because they could have gotten a capital U from their parents, or they could have gotten a capital U, little u from their parents. So we really don't know. We just know that they have at least one dominant gene. Okay, let's look at complex patterns of inheritance now. So we're on page 420. Oh, by chance, Mendel studied traits only influenced by one gene with 2 alleles. And that's significant, okay? That all of the traits that he studied just had one gene with 2 alleles. However, we know now that some inherited traits have a complex patterns of inheritance. There are different types of dominance, for example. Recall that for pea plants, the presence of one dominant allele produces a dominant phenotype. So if you have one dominant allele for purple, you're going to have a purple flower. However, not all allele pairs have a dominant recessive interaction. For example, incomplete dominance. Sometimes traits appear to be combinations of alleles. Alleles show incomplete dominance when the offspring's phenotype is a combination of the parents' phenotypes. For example, a pink camellia, as shown in figure 9, let's zoom down to here, looking at this one. Oops. All right, the pink camellia. Let's see. Results from incomplete dominance, a cross between a camellia plant with white flowers and a camellia plant with red flowers produces only camellia plants with pink flowers. So that means if you've got a true breed here and here, they both contribute something to the phenotype here, okay? So the phenotype shows that it has both a gene from a parent white plant and a gene from a parent red plant in order to create the offspring as pink. All right, what about codominance? Well, codominance, the coat color of some cows is an example of another type of interaction between 2 alleles, when both alleles can be observed. All right, so what's the difference between the 2? Well, the incomplete dominance is a combination. Okay, so it doesn't look like either one of them. Okay, that is the different, so this flower has some color in it, but it's based on, it's like a blending. Okay, it's a combination or a blending. With codominance, both can be observed, and that's kind of confusing, but I hope you'll understand it a little bit better when we look at these pictures. Sometimes you see flowers like this, like you might see a petunia that has like little stripes in it. Like maybe it's purple and white striped. Okay, so maybe it got its purple from one parent and it's white from another parent. So you can see both. It's not a blending. It's not a mixture, but you can actually see both. So in this example, if a cow inherits the allele for a white coat color from one parent and the allele for a red coat color from the other parent, the cow will have both red and white hairs. So you can actually see both. So let's look at that. So there's your white, your red, and they call this a roan coat color. So some parts of it are red and some parts of it are white. And so you can see both of the colors in the cow's coat. All right, another example of this codominance is our blood types. Oh, actually, no, that's not true. I'm going to stop that and take that off.